Hello, IB biology students. This is Miss Sheely. I am about to start the 1.5 Origin of Cells notes. I do ask that, as always, you listen, stop, and pause whenever needed. Make sure that you're recording your notes, whether you're using the PowerPoint or just your own blank sheet of paper. And then make sure you're also jotting down any questions you have so that we can discuss them when we review everything in class later. All right, so we do know now that cells can only be formed from pre-existing cells. At one time, though, we did believe that in spontaneous generation, and spontaneous generation is the formation of living organisms from non-living matter. It's that idea of poof, it just appears, and there's no precursor for that. There have been many different people over the years, most starting in ancient times, that have been able to document when some living organism has arisen, such as Theophrastus, who was a Greek philosopher, and he noted that a plant named Silphinum sprouted from the soil where there was previously no plants in that soil. Aristotle has documented many things, and some of them being that insects form from the feces of animals and that nothing was there except for the feces. Paracelsus is the spontaneous generation of mice from decaying matter. All of these gentlemen and many, many others have copious documentation of their thoughts on organisms arising from where there was no life before. But then we have a series of scientists who really have helped us to realize that cells can only be formed from pre-existing cells. And the first scientist is Francisco Redi. In his experiment, he took some jars and he put some meat in the jars. He left some open to the atmosphere and he covered others with gauze, and he let them sit for a period of time. And what he found was that the jars that were open to the environment had flies, whereas the jars that were not open, they were covered with gauze, did not have any flies develop. That wasn't quite enough for the scientific community and the community as a whole to really disbelieve this idea of spontaneous generation. So the next scientist that we have is Lazzaro Spallanzani, and his experiment, he took broth or gravy and he boiled it. He boiled it to remove any microorganisms. Well, they didn't know about microorganisms at the time, but to kind of purify the gravy. And then he left some in an open flask and some he closed the flask and then let that sit for a period of time. Of course, the open flask did develop things in it. They weren't sure what these things were, and the closed flask did not. That still was not quite enough. So the last scientist who's really helped us realize about that cells come from pre-existing cells is Louis Pasteur, and he did many, many experiments, but the one that most solidified or unsupported this idea of spontaneous generation was the one where he created several different curved necked flasks. And he took the flasks and, just like Spallanzani, he boiled his broth and let it sit, and nothing happened. Nothing formed. There was no living matter in the broth. And then after an extended period of time, he broke the neck off the flask, the special curved neck, and made it a shorter distance for the organisms or for life to enter. And what he found whenever he broke the neck off was that life did appear. So all of these things, plus other scientists, have led us to the idea that cells can only be formed from pre-existing cells. We do need to remember that a cell is a highly complex structure, and at this time we have no natural mechanism that has been suggested for producing cells from simpler subunits. That, in addition to no example is known of increases in the number of cells in a population, organism, or tissue without cell division occurring first, and that viruses are our exception, but although they are produced from simpler subunits, They do not consist of cells, and they cannot only be produced inside of host cells that they have infected. So all of these three things, plus our scientists who did our work against spontaneous generation, have led us to the realization that cells can only be formed from pre-existing cells. All right, at this time we're going to watch a little cell theory video. It doesn't go into so much of the three guys that we were just talking about, but it does kind of wrap up cell theory very nicely, and it will be a great review.
One of the great things about science is that when scientists make a discovery, it's not always in a prescribed manner, as in only in a laboratory under strict settings with white lab coats and all sorts of neat science gizmos that go beep. In reality, the events and people involved in some of the major scientific discoveries are as weird and varied as they get. My case in point, the weird history of the cell theory. There are three parts to the cell theory. 1. All organisms are composed of one or more cells. 2. The cell is the basic unit of structure and organization in organisms. And 3. All cells come from pre-existing cells. To be honest, this all sounds incredibly boring until you dig a little deeper into how the world of microscopic organisms and this theory came to be. It all started in the early 1600s in the Netherlands, where a spectacle maker named Zacharias Janssen is said to have come up with the first compound microscope, along with the first telescope. Both claims are often disputed, as apparently he wasn't the only bored guy with a ton of glass lenses to play with at the time. Despite this, the microscope soon became a hot item that every naturalist or scientist at the time wanted to play with, making it much like the iPad of its day. One such person was a fellow Dutchman by the name of Anton van Leeuwenhoek, who heard about these microscope doohickeys, and instead of going out and buying one, he decided to make his own. And it was a strange little contraption indeed, as it looked more like a tiny paddle the size of a sunglass lens. If he had stuck two together, it probably would have made a wicked set of sunglasses that you couldn't see much out of. Anywho, once Leeuwenhoek had his microscope ready, he went to town, looking at anything and everything he could with them, including the gunk on his teeth. Yes, you heard right. He actually discovered bacteria by looking at dental scrapings, which, when you keep in mind that people didn't brush their teeth much, if at all, back then, he must have had a lovely bunch of bacteria to look at. When he wrote about his discovery, he didn't call them bacteria as we know them today, but he called them animalcules, because they looked like little animals to him. While Leeuwenhoek was staring at his teeth gunk, he was also sending letters to a scientific colleague in England by the name of Robert Hooke. Hooke was a guy who really loved all aspects of science, so he dabbled in a little bit of everything, including physics, chemistry, and biology. Thus it is Hooke who we can thank for the term the cell, as he was looking at a piece of cork under his microscope, and the little chambers he saw reminded him of cells, or the rooms monks slept in their monasteries. Think college dorm rooms, but without the TVs, computers, and really annoying roommates. Hooke was something of an underappreciated scientist of his day. Something he brought upon himself as he made the mistake of locking horns with one of the most famous scientists ever, Sir Isaac Newton. Remember when I said Hooke dabbled in many different fields? Well, after Newton published a groundbreaking book on how planets move due to gravity, Hooke made the claim that Newton had been inspired by Hooke's work in physics. Newton, to say the least, did not like that, which sparked a tense relationship between the two that lasted even after Hooke died, as quite a bit of Hooke's research, as well as his only portrait, was misplaced due to Newton. Much of it was rediscovered, thankfully, after Newton's time, but not his portrait, as sadly no one knows what Robert Hooke looked like. Fast forward to the 1800s, where two German scientists discovered something that today we might find rather obvious, but helped tie together what we now know as the cell theory. The first scientist was Matthias Schleiden, a botanist who liked to study plants under a microscope. From his years of studying different plant species, it finally dawned on him that every single plant he had looked at were all made of cells. At the same time, on the other end of Germany was Theodor Schwann, a scientist who not only studied slides of animal cells under the microscope and got a special type of nerve cell named after him, but also invented rebreathers for firefighters and had a kickin' pair of sideburns. After studying animal cells for a while, he too came to the conclusion that all animals were made of cells. Immediately, he reached out via snail mail, as Twitter had yet to be invented, to other scientists working in the same field, and it was Schleiden who got back to him, and the two started working on the beginnings of the cell theory. A bone of contention arose between them, as for the last part of the cell theory, that cells come from pre-existing cells, Schleiden didn't exactly subscribe to that thought, as he swore cells came from free cell formation, where they just kind of spontaneously crystallized into existence. That's when another scientist, named Rudolf Virchow, stepped in with research showing that cells did come from other cells. Research that was actually, how to put it, borrowed without permission from a Jewish scientist by the name of Robert Remak, which led to two more feuding scientists. Thus, from teeth gunk to torquing off Newton, crystallization to Schwann cells, the cell theory came to be an important part of biology today. Some things we know about science today may seem boring, but how we came to know them is incredibly fascinating. So if something bores you, dig deeper. It's probably got a really weird story behind it somewhere.
All right, so I hope you enjoyed that. It was a nice little recap of some of the things that we've learned in this unit. All right, so there are some things about the origin of the first cells that we need to come to realize. So we know that cells come from other cells, but where in the world did the first cell come from? So one of the experiments that you need to be familiar with is an experiment by Stanley Miller and Harold Urey. They developed this apparatus that you can see here. It's a closed apparatus, and what they did was they put they boiled water, and that rose into steam, and then they passed that steam through a mixture of methane and ammonium and things that were thought to be representative of the early atmosphere of Earth. And then they stimulated that chamber with electrical discharges, and that was to simulate lightning. They allowed that to be condensed or cooled so that it reformed into water and then fell back to the earth. And then it continued through this closed loop. And what they found was that when they took a sample here, they did find that there were some amino acids and other compounds that were produced, and that these amino acids and other carbon compounds could easily be used to make the first molecules that would be the beginnings of cells. Another idea of the origin of the first cells is deep sea vents. This is a possible site for the origin of the first carbon compounds. Deep sea vents are just cracks in the Earth's surface that gush hot water. In that hot water, they also carry inorganic chemicals, and these chemicals represent supplies of energy that could assemble into carbon compounds or polymers. Another idea for the origin of the first cells is the formation of membranes. Phospholipids and other lipids naturally assemble into bilayers when they're placed in solution, and these bilayers readily form into vesicles that resemble plasma membranes of a small cell. So you can see here, this is a micelle where the phospholipids are all pointing towards each other, so there is just water. This micelle would be in a fluid environment in a solution. A liposome, we have a bilayer that is produced so you can have water on the inside as well as water on the outside, which would be the idea behind the phospholipid bilayer. So this would allow for different internal chemistry from the surrounding environment if we were able to get to this liposome arrangement. The last thing that we need is we need a mechanism for inheritance. So living organisms currently have genes that are made of DNA, and we use enzymes as catalysts to undergo all the reactions in our cells and in our bodies. Well, the problem is to replicate the DNA, we need enzymes. But to make enzymes, we need genes. So we have this chicken and the egg conundrum. What came first? And the current thought is that, well, RNA is also a genetic material. The benefit of RNA is that it can self-replicate itself. but it can also act as a catalyst to make other proteins from it. And there is thought that perhaps that RNA was the first method or mechanism for inheritance in cells, that DNA has evolved from the RNA. So those are the 4 ideas behind the origin of the first cells. So now we have a prokaryotic cell. Well, how did we get eukaryotic cells? Well, this is an idea called endosymbiosis. So we have a cell, and in that cell, we have a cell membrane or a plasma membrane. We have a fluid environment, and we have that genetic material, whether it be DNA or RNA. And over time, that cell starts to fold. And within those folds, we get endoplasmic reticulum and maybe even a nuclear membrane or nuclear envelope develops. So now we still have the beginnings of a much more complex system. Well, mitochondria were also once free-living prokaryotic organisms. Then a larger prokaryotic organism or cell ingested the mitochondria by endocytosis. Since the larger cell could aspire anaerobically, but now the mitochondria can respire aerobically, it didn't break down the smaller organism. It allowed the smaller organism to live inside of it and produce its own energy. And if we remember from our introductory biology, aerobic respiration produces much more energy than anaerobic respiration. So this became a mutualistic relationship between the much larger prokaryotic cell and the much smaller mitochondrial prokaryotic cell. Over time, natural selection favored this mutualistic relationship as the cell was more efficient no matter what type of environment it was in. The same thoughts are applied to the idea of the chloroplast, that the chloroplast was also once a free-living prokaryotic organism, that it was also engulfed by a much larger prokaryotic cell, and then over time that prokaryotic organism was just turned into or emerged as an organelle to support the larger cell. All right, guys, so that is a brief rundown of the origin of cells. This is a short little section. I hope that you jotted down several questions that you have. Please be willing to share those in class, and have a great evening. Thanks.